Okay, so welcome back to this next video in which we are discussing cholesterol metabolism. Okay, right. So we're in the process of discussing intracellular cholesterol homeostasis, so how peripheral cells and hepatocytes control the amount of cholesterol they have. Okay, and we've just been discussing biosynthesis of cholesterol, which is a pathway which can occur in both hepatocytes and in peripheral cells. And we've specifically been looking at how the synthesis of cholesterol can be controlled by controlling this step here, which is the conversion of HMG-CoA to mevalonate, which is catalyzed by this enzyme HMG-CoA reductase. Okay, and we've discussed the way that you turn cholesterol synthesis off in cells which have a lot of cholesterol is that the HMG-CoA reductase enzyme will be destroyed basically when there is too much cholesterol within the cell. Okay, and we've discussed that HMG-CoA reductase is this enzyme that sits in the ER lumen, and I want to make a correction here. I called this the sterol sensing domain, but then wrote sterol sensing cholesterol. Okay, so this should have said sterol sensing domain here. Okay, so basically we've discussed how cholesterol can bind to this sterol sensing domain when it's in too high a concentration in the ER membrane, which is indicative that the cell has too high a concentration of cholesterol. Okay, this will trigger a conformational change in this transmembrane domain of the HMG-CoA reductase enzyme, and it then binds to a protein called Insig-1 or another protein called Insig-2, which both do the same thing, and therefore we're denoting this Insig-1/2, which then results in the HMG-CoA reductase being destroyed by the ubiquitin proteasome pathway. So the cell will destroy its HMG-CoA reductase enzymes when cholesterol level within the cell is high, okay, and therefore will stop synthesizing more cholesterol when cholesterol is high. Okay, right. What I now want to discuss is the SREBP pathway, which is the pathway by which you can respond to the cell's cholesterol level being too low. Okay, and one of the key responses that you're going to induce is the expression of new HMG-CoA reductase enzymes. So you're going to make new HMG-CoA reductase enzymes to replace those ones that have been lost. Okay, right. So let's now turn our attention onto the SREBP pathway then. Okay, so let's start by discussing the SREBP protein itself. So firstly, what does its name stand for? Well, its name stands for the SRE, and I will tell you what the SRE actually stands for in a moment. And then the BP is just for binding protein. Okay, so a huge number of proteins have BP in their name. Again, all it stands for is binding protein. So SREBP stands for SRE, and we'll come to what that stands for in a moment. Binding protein, and that's the BP. Okay, so now what we want to ask then is, what does SRE stand for? Okay, well, SRE stands for the sterol response element. Okay, which is a certain sequence of organic bases in DNA, basically, okay, which a certain portion of these SRE binding proteins is capable of binding to and acting as a transcription factor. Okay, so sterol response element is what SRE stands for. Okay, right. So let's start off by discussing what the structure of an SREBP protein is, and then we'll have a look at the basic overview of what the SREBP pathway actually is and what it does, and then we'll look at it in more detail as to how it actually occurs. Okay, so SREBP proteins then, they are once again a protein that is in the ER membrane. Okay, so in the endoplasmic reticulum membrane, they're an integral membrane protein in the ER membrane. So I'll once again draw out the ER membrane here. So this is the endoplasmic reticulum membrane here. Okay, and once again we'll let the portion above this membrane represent the cytoplasmic side. So this is the cytosol. Okay, and the portion below will represent the luminal side. Okay, right. Now what I want to do is show you the structure of the SREBP proteins sitting in the ER membrane. So basically SREBP proteins have two membrane spanning alpha helices, and again they are a single protein, okay? And both their N and C terminals are going to be on the cytoplasmic side of the ER membrane. So here's the N terminal. Then they're going to have a really important portion, which is the portion that's actually capable of acting as a transcription factor. Okay, and this portion is called the BH-LH domain. Okay, and that refers to the type of transcription factor. Okay, so basically this stands for basic helix, that's the BH, and then loop helix. Okay, and that describes the structure of this domain basically. Okay, it consists of a alpha helix which has lots of basic residues in. Then you have a loop, and then you have another helix. If I just draw a picture here, here's the basic helix, then you'll have a loop, and then you'll have another helix here. Okay, so that domain will contain this basic structure. Okay, and I wasn't using basic in the same context as basic here means. I was meaning basic as in simple or foundational. Okay, so that's our basic helix there in blue. Here is our loop in green here. Okay, and then we've got our other helix here in red. So this domain will have a structure like that basically, and many transcription factors have this kind of structure, and that's how they bind to DNA basically. So BH and H transcription factors, it's a common type of transcription factor. Okay, and it just refers to the structure of the protein and how it then binds to the DNA. Okay, right, so I'll colour in that BH and H domain here in orange. Okay, now at the moment this BH and H domain is not going to be binding to DNA at all because it's attached to the rest of the protein, which is anchoring it in the ER membrane, so it's not going to be doing anything at the moment with regards to affecting gene expression. Okay, so here are the two transmembrane domains of our SREBP protein. Then you have an important domain at the other side of the protein, which I'll colour in blue here. Okay, and this is known as the regulatory domain of the SREBP protein. Okay, so in blue here, this is the regulatory domain. Okay, and this portion is going to associate with another protein called SCAP, which we'll meet in a moment. Okay, so that's the basic structure of an SREBP protein sitting in the ER membrane, and I think I'll just colour in those two membrane spanning alpha helices in yellow. Okay, right, so now what I want to do is give you a basic overview of how the SREBP pathway actually occurs. Okay, so basically the SREBP pathway is the cell's response to low cholesterol. Okay, so the entire pathway begins with cholesterol being low. Okay, now, when cholesterol is low, SREBP proteins are going to get cut up, and we will look in a lot more detail at exactly how this occurs in a moment. But the crucial thing is that you're going to chop off this BH and H domain here of the SREBP protein from the transmembrane domains which anchor it attached to the membrane basically. Okay, and you'll produce this little fragment here. So here's the N-terminus. Here then is our BH and H domain here. Okay, and then it will have this little portion here as well. So I'll just colour this in orange. So when cholesterol is low, SREBP proteins get cut up. You produce this little fragment of the SREBP protein, which has a special name. This little fragment is now known as the N-SREBP. Okay, and the reason it's called the N-SREBP is because it's going to go into the nucleus. Okay, so the N here stands for the nuclear SREBP. Okay, so it's the portion that is actually now going to go into the nucleus and affect gene expression. Okay, so now once you cut this portion off the rest of the SREBP protein, which is keeping it attached to the membrane, it's now going to go firstly into the cytoplasm, then it's going to go into the nucleus. Okay, in the nucleus it's then going to dimerize, it's going to find the body and it's going to dimerize with it. Okay, and that dimer of these two NSREBP proteins is then going to bind to the sterile response element, which is this sequence of DNA that is found in the gene control regions upstream of many genes, and which will promote or repress the gene expression of the downstream genes. So it will change gene expression basically. Okay, so if I just show this, so here now, this pair of parallel lines here, this is now no longer representing a phospholipid bilayer, but instead is representing a piece of double-stranded DNA. So let's say here is a gene that's going to be controlled by cholesterol level. Okay, so this is some gene here. Then in its gene control region, which is the portion upstream of the gene which controls how likely it is to be transcribed, you're going to have a little sequence of a few organic bases. Usually these sort of sequences for controlling gene expression are no more than 10 base pairs. I don't know the exact length or sequence of the sterol response element, but it will be around 10 base pairs. Okay, so this will be this SRE, this sterol response element, and it's just a sequence, a conserved sequence of organic bases basically. Okay, and now what can happen is this dimer of nuclear SREBPs can now bind to this sterol response element. So this is a dimer of these two portions here. Okay, so both of these are these nuclear SREBPs, sterol response element binding proteins. Okay, and now what that will do is it will change the level of transcription of the downstream gene in some way. Okay, and I won't go through the details of how it can actually affect the transcription of the downstream gene, because it takes me quite a long time to explain all of that. Okay, but it will change the expression of the downstream gene. It will either increase it or it will decrease it basically. Okay, and two of the absolutely key ones that we're interested in is that the NSREBP can bind to sterol response elements upstream of genes for, firstly, the HMG-CoA reductase enzyme, okay, and it will activate the expression of that gene. So you'll produce more HMG-CoA reductase, which means that you can start up your cholesterol biosynthesis again. Okay, so cholesterol biosynthesis can go back up. Okay, so that's one of the key things that you're going to upregulate the expression of when these nuclear SREBPs go into the nucleus and change gene expression. Another thing that you're going to upregulate on the within the cell is something called the LDL receptor. Okay, so low density lipoprotein receptor. Okay, and these receptors will be put in the cell membrane, and basically they can be used to endocytose LDL particles, and therefore they can be used to gain cholesterol from the LDL pool in the blood basically. And again, this is used in both peripheral cells and hepatocytes. Okay, and we will be discussing the LDL receptor in a lot more detail later on. Okay, we'll discuss exactly how it can endocytose LDL particles and how that can provide cholesterol to the cells. Okay, right. So firstly then now, I want to discuss this initial portion in more detail. How does low cholesterol cause the cleaving of the SREBP proteins to produce these nuclear SREBPs? Because it's not quite as simple as you might think. It is a quite elaborate little pathway. Okay, right. So firstly, let me talk about how when you have high cholesterol, you don't get the cleavage of SREBP, and then I'll talk about what happens when you have low cholesterol and how then things change and you do get the cleaving of SREBP. Okay, so the first thing that we need to talk about is that SREBP, when it's in the ER membrane, it is complexed with another protein. Okay, and this is true whether you have high cholesterol or whether you have low cholesterol, basically. Okay, so this next protein that I want to talk about is a protein called SCAP. So now what I'm doing is I'm drawing out the ER membrane again. So these two parallel lines have gone back to representing the membrane rather than a piece of double-stranded DNA. Okay, and let's firstly just draw out our SREBP protein once again. So here's that BHLH domain. Here are our two transmembrane domains, and now we're going to see the function of this regulatory domain here. Okay, and there's the C-terminus of our protein. So let's just colour these portions in. So we'll have the BHLH domain here, once again in orange. We'll have the transmembrane domains, once again here in yellow. Okay, and we'll have the regulatory domain here in blue. Okay, so once again, I'll just stress that this is the cytoplasmic side of the membrane, this is the luminal side of the membrane. Okay, so now what's going to happen is SREBPs that are in the ER membrane, they end up complexing with this other protein, which is known as SCAP. Okay, so what does SCAP stand for? Well, the S is for SREBP. Okay, so it's the SREBP cleavage activating protein. So the C is for cleavage. Okay, the A is then for activating. Okay, and then finally the P is for protein. So SCAP is the SREBP, the sterol response element binding protein, cleavage activating proteins. It's a massive great name. Okay, so SCAP is another protein which is present within the ER membrane and which is capable of binding to SREBP proteins and indeed will bind to SREBP proteins. Okay, so now let me show you the structure of SCAP. Now I'll start from this side, but this side is actually the C-terminal of SCAP, okay? So we're starting at the end of the protein basically. And SCAP basically has a domain which is capable of binding to the regulatory domain of the SREBP protein. And I'll colour this domain in red here. Okay, then you'll have 8 membrane-spanning alpha helices, similar to the HMG-CoA reductase enzyme. Okay, and then you'll have the N-terminus here. Okay, right. Now, the other thing about SREBP cleavage activating protein, SCAP, that is similar to the HMG-CoA reductase, is that it also has a sterol sensing domain here in red. Okay, so those 5 membrane-spanning alpha helices, number 2, number 3, number 4, number 5, number 6 here, once again, those form a sterol sensing domain of this SCAP protein. Okay, so this is a sterol sensing domain. So this binding of SCAP to SREBP, this occurs whether cholesterol is high or low. However, what I now want to discuss is what happens when cholesterol is high, and how what happens when cholesterol is high is going to prevent the breakdown of SREBP, and then we'll talk about what happens when cholesterol is low and how that's going to promote the breakdown of SREBP. Okay, so firstly, let me talk about what happens when cholesterol is high. So, when cholesterol is high, you can probably guess what's going to happen. We've got lots of cholesterol in the ER membrane, that means. Okay, so if the cell has lots of cholesterol, you end up with lots of cholesterol in the ER membrane. Cholesterol is going to bind to the sterol sensing domain of the sterol response element binding protein, cleavage activating protein here. Okay, what does that then trigger? Well, it triggers the SCAP protein to then complex with another protein, a third protein, and we've seen this protein before. Okay, so I'll add this protein in here over the top of those cholesterol molecules there. Okay, and this is Insig 1 or Insig 2. Okay, so basically, when cholesterol is high in the cell, cholesterol is going to bind to the sterol sensing domain of the SCAP protein, which is in complex with the SREBP protein. Okay, and that's going to cause the SCAP protein to change conformation so that it can now bind to Insig 1/2 proteins in the ER membrane, which we know are present in the ER membrane because we've met them before. However, Insig 1/2 binding to SCAP causes a very different thing to what it did with HMG-CoA reductase. Okay, what it now does is it traps this complex of SREBP and SCAP in the ER membrane. It stops it moving from the ER membrane, and that's really important because for the SREBP to be cleaved, it has to move to the Golgi basically. It can't be cleaved in the ER membrane. To be cleaved up, it has to go to the Golgi membrane. Okay, so when there is high cholesterol basically, cholesterol is going to bind to the SCAP, and SCAP will then bind to Insig 1/2, and Insig 1/2 holds this SREBP SCAP complex in the ER membrane basically, and stops it from moving to the Golgi membrane. Okay, right, so that's how you stop the breakdown of SREBP when cholesterol is high basically. Okay, now let's talk about what's going to happen when cholesterol is low. So when cholesterol is low, the sterol sensing domain of the SCAP protein doesn't have cholesterol bound to it. Okay, so it doesn't bind to Insig 1/2, and now you end up just with the SREBP bound to the SCAP protein. So you end up with this SREBP SCAP complex on its own with no Insig 1/2. And basically, when SREBP and SCAP are complexed without the Insig 1/2, the whole thing gets moved from the ER membrane to the Golgi membrane. Okay, so let me just draw a little picture here. Okay, so if we've got our cell here, like so, here is the nucleus, okay, and we'll have the outer nuclear membrane here, and then of course the outer nuclear membrane is continuous with the ER membrane, and indeed the ER lumen is continuous with the space in between the inner nuclear membrane and the outer nuclear membrane. Okay, so here is our endoplasmic reticulum basically, that's what I've drawn here, this is the ER. Okay, and then of course we'll have the Golgi apparatus, which is this stacked bunch of cisternae here. Okay, and basically what we're now going to do is we're going to move this SREBP SCAP complex from being within the ER membrane to being within the Golgi membrane here. Okay, and basically the way that will be done is you'll pinch off little vesicles of the membranes. If I draw the picture larger here, that can be the ER membrane here. Okay, and here is the Golgi membrane here in blue. What you're going to end up doing is pinching off little vesicles of the ER membrane, like so, and then transporting these vesicles to the Golgi and then refusing them with the Golgi membrane basically. Okay, and that's how we can deliver the SREBP SCAP complexes to the Golgi membrane. So now let's say that's happened because cholesterol is low within the cell. Okay, so now I'm drawing a picture of the Golgi membrane here. Okay, so once again, this can represent the cytoplasmic side, okay, above, and this can represent the lumen of the Golgi side. Okay, so this side is facing into the cytoplasm here, and this side is facing into the lumen of the Golgi. Okay, and this is the Golgi membrane. So now what we've got is the SREBP SCAP complex in the Golgi membrane, and it will be oriented the same way as it was in the ER membrane, i.e. it will be oriented with the BHLH domain of the SREBP protein facing into the cytoplasm rather than facing into the lumen. Okay, so the picture is now going to look very similar. So once again, here is the BHLH domain of our SREBP protein here. Okay, then we'll have the two membrane spanning alpha helices of our SREBP protein. Here is the regulatory domain here, and then the C-terminal here. Okay, so we'll colour these portions in. Here is the BHLH domain in orange, the two membrane spanning alpha helices here in yellow. Okay, here's the regulatory domain here in blue. Okay, and that will still be in complex with that domain of the SCAP protein, which is capable of binding to that regulatory domain. So I'll just show this here. So here's the C-terminal. Here now is our 8 membrane spanning alpha helices, and then there's the N-terminal. Okay, so basically, when cholesterol is low, you move the SREBP SCAP complexes from the ER membrane to the Golgi membrane, whereas when cholesterol is high, you don't, because when cholesterol is high, cholesterol binds to the sterol sensing domain of the SREBP cleavage activating protein SCAP, and that then binds to Insig 1/2, which maintains the SREBP SCAP complex in the ER membrane. Okay, right. So we now have this SREBP SCAP complex in the Golgi membrane. Now what happens? Well, basically also in the Golgi membrane, and unfortunately I'll have to show this separately because there isn't enough room on that picture there. Also in the Golgi membrane, you're going to have two important enzymes, which again I'm just going to show as blobs, but these are enzymes that are integral membrane proteins in the Golgi membrane. Okay, and these two enzymes are called the Site1 protease, which for short is abbreviated to the S1P, and the Site2 protease, which for short is abbreviated down to the S2P. Okay, so let's colour code these in. So we'll have S1P in green here, and I'll just write out its full name. Okay, so the S is for site, then the 1 is for 1, and then the P is for protease. Okay, and similarly for site 2 protease, the S is for site, the 2 is for 2, and then the P is for protease. Okay, so basically these are enzymes which are present in the Golgi membrane, which are capable of breaking down the SREBP. So you now hopefully see how this is so crucial. So the enzymes for breaking down the SREBP protein are not in the ER membrane, they're in the Golgi membrane. So if you want to break down SREBP, which we want to do if cholesterol is too low, you have to bring the SREBP to the Golgi membrane so that the site 1 and site 2 proteases can act on it, and that's exactly what is going to happen when cholesterol is too low within the cell, basically. Okay, right, so now what do the site 1 and site 2 proteases do? Well, let me firstly show what the site 1 protease does. So site 1 protease cuts the SREBP at this point here. Okay, so between the two transmembrane domains. Okay, and that will produce two separate fragments. You'll end up with the N-terminal with the BH&H domain, and then a single membrane-spanning alpha helix, and then you'll end up with this second portion which will still be attached to SCAP here. Okay, so let me now show this. So here is our N-terminal portion of SREBP here. Here is that BH&H domain here, and now we've got a single membrane-spanning alpha helix still anchoring us to the Golgi membrane. However, that's not complete yet. We haven't got the nuclear SREBP yet. It needs to be cut again. Okay, so the site 1 protease goes first to produce this thing, and then the site 2 protease will have its go afterwards. So it will cut the SREBP at just the starting point of the transmembrane domain here, the first membrane spanning alpha helix, and then you'll actually get this nuclear SREBP being produced, basically. Okay, and that's now free in the cytoplasm. It can go into the nucleus where it will dimerize, and there it will bind to steroid response elements that are present in the gene control regions of many genes, and it can influence the transcription level of those genes, and thereby it can change the epigenetics of the cell. It can change gene expression within the cell. Okay, and the two key targets that we're going to talk about are the fact that it will upregulate HMG-CoA reductase, and we know what that will do. That will boost up cholesterol biosynthesis, exactly what we want if we've got too little cholesterol within the cell. Okay, and it will also now increase the expression of this LDL receptor, okay, which is a new guy. We haven't seen that yet. Okay, which is now going to lead to the endocytosis of LDL particles from the blood into the cell, basically, and that's going to be another way that the cell can get cholesterol into itself when it's got too little cholesterol. And it is the LDL receptor and the LDL receptor mediated endocytosis of LDL that we will be turning our attention to in the next video.